All right, today we are going on a journey. We're stepping back in time into a quiet monastery garden to solve one of the biggest mysteries of life itself. We're going right back to the very beginning to figure out how the science of genetics even started.
I mean, really think about it. It's a question people have been asking for thousands of years. You see it all the time, right? A child has their father's nose or their grandmother's hair. But how? What's actually going on behind the scenes? For centuries, it was a total puzzle. Today, we're going to put the pieces together.
So here's our game plan. We'll start with that ancient mystery, then we'll meet the monk who used simple pea plants to start finding answers. We'll see how he cracked the code and discovered the two fundamental laws of inheritance. Then we'll look at how we can use his work to make predictions, and finally, how it all connects to the incredible world of modern genomics. Let's dive in.
Okay, so our story really begins with something everybody has noticed. For as long as we've had families, we've known that kids tend to look like their parents. But the actual how and why of it, that was a complete black box.
The big idea we're chasing here is called heredity, or you might hear it called inheritance. It's just the official term for how traits get passed down from one generation to the next. This is the phenomenon that scientists were desperate to understand, and for the longest time, they really had no clue how it worked.
And then, the first huge breakthrough came from a place you would never expect, a monastery garden. And our detective in this story, he was an Austrian monk with a brilliant scientific mind and, it turns out, a whole lot of patience.
Meet Gregor Mendel. Back in the mid-1800s, through some incredibly careful and detailed experiments, he started to figure out the basic rules of heredity. His work was so important, so foundational, that today we call him the father of genetics. He literally wrote the first chapter.
Now, part of Mendel's genius was picking the perfect subject to study. So, why pea plants? Well, they were perfect for a few key reasons. First, they had really simple, observable traits. It was either or. Seeds were yellow or green. Flowers were purple or white, no in between. They also grew super fast, so he could get a ton of data in a short amount of time. Plus, he could control exactly which plants mated with which. And, most importantly, he could start with what are called true breeding plants. Plants that, generation after generation, always produced identical offspring. This gave him a clean, reliable starting point.
So, with his perfect subject ready to go, Mendel started his experiments. And this is where the clues that would completely change biology started to pop up. His first move was pretty straightforward. He took a true breeding plant that always made yellow seeds and crossed it with a true breeding plant that always made green seeds. He called this first group the P, or parent generation. Simple enough, but what happened next was not simple at all. Every single one of the baby plants, what he called the F1 generation, every single one had yellow seeds. The green trait was just gone. Poof. It was like it had been totally erased. So where did it go? This was the first big clue in the mystery.
So Mendel took it a step further. He let these new F1 yellow seed plants pollinate themselves. And in the next generation, the F2 generation, bam! The lost green trait suddenly showed up again. This was the huge aha moment. The trait wasn't destroyed, it was just hiding.
But here's the part that really changed everything. The green trait didn't just come back randomly. It came back in a beautifully predictable pattern. For every one green seed plant, there were about 3 yellow seed plants. This consistent 3-to-1 ratio was the mathematical key that unlocked the whole puzzle. Mendel knew there had to be a rule behind this.
To explain this, Mendel proposed that for every trait, an organism gets 2 factors, one from each parent. Today, we don't call them factors, we call them alleles. And an allele is just a specific version of a gene. So for the seed color gene, there was a yellow allele and a green allele.
And this is what explained the vanishing act. Mendel figured out that one allele must be dominant and the other recessive. The dominant allele, which we show with a capital Y for yellow, is like the loud one in the room. If it's there, you're going to see its trait. The recessive allele, lowercase y for green, is the quiet one. It only gets to show its trait if there's no dominant allele around to mask it.
Okay, so this brings us to 2 more vocab words we need. If a plant has 2 of the exact same alleles, like 2 yellows, YY, or 2 greens, YY, we call that homozygous. But if it has 2 different alleles, like 1 yellow and 1 green, YY, we call it heterozygous. So you see, all those F1 plants were heterozygous.
And this leads to a really important distinction, the difference between genotype and phenotype. The genotype is the actual genetic code, the combination of alleles, like YY. The phenotype is the physical thing you actually see with your eyes, like yellow seeds. That's why a YY plant and a YY plant both have the exact same yellow phenotype. The dominant allele is running the show.
So, armed with these new concepts, Mendel was finally able to lay out two fundamental laws of inheritance that solved the mystery and basically built the foundation for all of modern genetics.
His first rule is the law of segregation. It sounds complicated, but it's pretty simple. It just means that when a plant makes its reproductive cells, like its pollen or egg cells, its two alleles for a trait split up. They segregate. So each reproductive cell only gets one of the two alleles. A YY parent can pass on a Y or a Y, but never both in the same cell.
His second rule, the law of independent assortment, is even cooler. It answers what happens when you look at more than one trait at a time, like seed color and seed shape. This law says that the alleles for different traits get sorted into reproductive cells independently of each other. In other words, just because a pea inherits the allele for yellow color doesn't make it any more or less likely to inherit the allele for a round shape. They're not connected. They're sorted independently.
So what's so great about these laws? Well, they don't just explain what happened in Mendel's garden. They give us the power to predict the future, genetically speaking. And one of the most powerful tools for doing that is something called a Punnett square.
Let's look at a simple one. This is a monohybrid cross, just looking at one trait. You put one parent's possible alleles on top and the others on the side. Then you just fill in the boxes. Here, we're crossing a heterozygous black guinea pig with a white one. You can clearly predict that half the offspring will be BB and black, and the other half will be BB and white. It's a perfect 1-to-1 ratio. It's an amazing predictive tool.
Okay, pop quiz. Let's see if you've got it. If a dominant trait, like non-curled ears in a cat, shows up in the kids in a 3-to-1 ratio, what does that tell you about the parents? That 3-to-1 ratio is the classic signature of one specific cross, crossing two heterozygous individuals. Both parents must have been heterozygous for that trait. It's the only way to get that result.
And that really brings us full circle. It's just incredible to think that these simple rules, figured out by a monk patiently counting thousands of pea plants, were the absolute starting point for everything we know about genetics today, from understanding inherited diseases all the way to mapping the entire human genome.
Just think about that. From a simple garden, the fundamental rules for all of life were written. It's just a powerful reminder that sometimes the biggest discoveries can come from the most humble and unexpected places. Thanks for joining us.